model of how acute stress will produce post-traumatic stress-like symptoms, at least to the extent that we can model that in a rodent. And all the data I'll talk about is done with rats in these studies. So I don't need to really remind the audience here that about some fraction of the individuals exposed to a traumatic event will go on to develop the psychological and physiological symptoms of post-traumatic stress disorder. What I want to do is just go through a few PTSD symptoms. Not that it's an exhaustive list, but these are the things that I think are particularly relevant to the model I'm talking about, and the things that we can model in the rat, because certainly we can't model everything in a rodent. So one is fear and avoidance of things that are associated with the traumatic event. The symptoms must be present for at least 30 days after the trauma. There's high comorbidity with other anxiety disorders. And one thing is that there's an increased propensity to acquire new phobias, even those not related to the original trauma. And that's something that this model is very especially relevant to. It tends to be resistant to exposure-type therapies. And the point I'm going to make is that there's a component, I think, of post-traumatic stress that's basically irrelevant to exposure therapy. So exposure therapy hits on one aspect, but it leaves the aspect that I'm going to talk about unaffected, and I'll show data to that effect. There's increased arousal, hypervigilance, exaggerated baseline startle responses, high rates of comorbidity between PTSD and alcohol abuse and alcohol dependence, and high rates of comorbidity between PTSD and depression. These are all things that in our model we have data relevant. I'm going to not talk about all of our data because it's just too much, so, but we've touched upon, I think, each of these, and I'll hit upon some of those today. So if we step back and say from a sort of a neurobiobehavioral perspective, what can happen during a single traumatic experience that's going to produce the lifelong symptoms that can characterize PTSD? I think there are really two components we can look at from a neurobiological perspective. And one is Pavlovian fear conditioning, and that's an associative phenomenon in that the cues that are present at the time of trauma provoke fear memories and responses. So in the laboratory, that's modeled by Pavlovian fear conditioning. Maybe you take a tone, pair it with an electric shock, and that specific tone will now produce fear responses. So it's associative in that nature. That's really currently the major focus of what's going on with biobehavioral research. So neuroscience research has really these days focused on this associative learning, this Pavlovian fear conditioning. And one thing is that this is treatable by exposure therapy, extinction-based type therapies. And there's a lot of great work going on in multiple labs, including my own, of how to try to make this better. That's not what I'm going to touch on today, though. What I want to touch on is something called stress sensitization that's non-associative. And one of the things about this is because it's non-associative, it's not locked in, it's not specific to the cues or the contexts originally associated with the trauma, that it can go beyond those original contexts. And for this, I think we don't have a treatment. There's no known treatment for this sort of thing. So what I want to do is advance the hypothesis today that many of the symptoms of PTSD are derived from this form of sensitization, and it's something that we're calling in the model stress-enhanced fear learning, or CEFL, and I'll show you why we call it that in a moment. In terms of the organization of my talk, what I want to do is first start out with the behavioral phenotype. I'll show some old data, some recent data that just describes the basic behavior. Then some mechanisms of what is required to induce these changes, what is required during the stress, what has to happen during the stress to produce the phenomenon, and then what are some of the potential mechanisms of expression. That'll be the end of my talk, a little more speculative, but I think it gives us some insight into what's going on that produces these symptoms. So here's the basic kind of model, and it's this is sort of the stripped-down version. I'm going to take you through different versions of it, but the core phenomenon is basically illustrated here. And in one day, on day one, we put the animal in a particular context, and what I call a context, it's a particular chamber that looks a certain way. It has a particular smell, a particular grid floor, particular background lighting, everything we can make about different about it is different from the other contexts. But so they go into one context on day one, and during that time, over the course of 90 minutes, they get 15 shocks. For your shock aficionados, it's a 1 milliamp, 1 second shock. They happen randomly over this time. There's nothing the animal can do about it. They're unpredictable over this 90 minutes. So that's our stressor there. And then what we'll do is on some other day, let's call it day 2 of the experiment, the animal will go into an entirely different place. So we eliminate as many characteristics between the two, make it as different as we can, and the animal receives one shock there, so a mild stressor. In this study, it's going to be the same intensity and the same duration as that shock, but it's only one shock. And on the third day, we'll measure fear of this environment that has one shock. And one thing about fear conditioning, so I'm doing context fear conditioning. One thing about fear conditioning you have to recognize is it's graded. If you use a mild shock, there's less conditioning than if you use a strong shock. If you use more trials, there's more conditioning than less. So it's graded. And my idea there is that the importance of that is that fear needs to be appropriate to the situation, right? It has to be graded to the degree of threat. If there's a mild threat here, like a bee comes in here, I want to have increased vigilance. If a tiger walks in here, I want to jump underneath the table there. But if I did that to a bee, it would be, not exactly right. So you want to have fear that's graded to the situation, and that's appropriate. But when you have fear that's disproportionate to the situation, that's when it's inappropriate. That's when we have a problem. So in this experiment, again, they got 15 shocks on this day. And what I want to point out is when they first go into this context, before anything happens, there's no fear. We use freezing as our measure of fear. Rats are very nice. They tell us how afraid they are by going into a motionless, crouching posture. They don't move. Everything stops. Even the vibrissae stops. We can score it by a human observer. We can have computers score it for us. But when they go into this other context, before anything happens, they don't freeze in there. There's not a generalization of the fear from this context to that context. Now, they do get a shock here. What happens the next day when we bring them back to this context? And here are the data for that. So what we see is animals who had no shock on this day here. They don't freeze very much. Animals who had a single shock but did not have the prior stress, did not have the 15-shock stressor, freeze somewhat. So they have a fear that's proportional to the one shock they received here. But these are the interesting animals, and it's this behavior here that's interesting. The animals who had the prior stress, even though they're not generalizing from the other context, show this potentiated or this enhanced fear. And so this is what we call stress-enhanced fear learning. And that's going to be my core measure of what's going on. I'll show you some other measures, but this is the main measure of what we have. A fear that occurs that's really disproportionate to the amount of stress you experienced in that situation. And again, we can do lots of different variants on this. In this experiment here, I use the same shock in both situations, the single shock, and there are theoretical reasons for doing that. But if I want to really make the argument that this is an enhanced fear, that it's disproportionate, what happens if you use a really mild stressor? So in this study here, they got the same 15 strong shocks as in the other study, but when they were brought to that new context, they received a single shock, some of them the same shock they received before, but others received a milder shock. And so here are the data from the test of the one-shock context where they got different groups got different shocks. The animals are split between males and females here. And the first thing you notice is that as that one shock gets stronger, even in the animals that had no prior stress, that there's more conditioning, the stronger shock. So conditioning is graded to the amount of threat that they received in the situation. But at every intensity here, what we see is that the animals had the prior stress have an exaggerated fear reaction. Yes, it's graded, but it's still exaggerated if they had the prior stress. What's particularly remarkable to me about this is that this really weak shock was not enough to support conditioning on its own in animals that had no prior stress. They're not freezing. If they had the prior stress, they show a strong reaction. We do not see a sex difference here. We see a sex difference at the high intensity between the amount of fear conditioning, but at least at the level of stress we're invoking, we're not seeing male-female differences there. And we can talk about that during questions if you like. So again, this prior stressor really gives you a disproportionate stay. They're just going to get this very short burst of a very loud white noise. So psh is white noise, and they get a burst of that. And here's their startle response. So here's the burst of white noise, and we're looking at their movement here. So when the noise occurs, that's the little blue shading here, everybody shows a startle response, but you can see that the prior stressed animals have an exaggerated startle response. So as in post-traumatic stress, we see enhanced startle reactivity. We see enhanced startle reactivity in this case as well. If we look at them over the time that they're in here, over the few minutes that they're in here, what we see is after that startle stimulus, there's a little bit of freezing in the animals who never had stress before. Just the only thing that happened to them bad was that loud noise. But the animals had the prior stress are showing a lot of freezing, a lot of this fear response after that loud noise. Now what we're going to do is bring them back the next day to this place where they had the loud noise, no noise, so it's did the loud noise condition in that situation. And here are the data for that. So again, not much freezing in the animals who just had the burst of loud noise but didn't have the prior stress. If you had the prior stress, now we see this enhanced conditioning. This noise is able to condition fear to that environment in those animals. It's certainly, if you look, the scale has changed here. It's not as much as if I gave a shock, but something that's relatively innocuous here is conditioning a pretty pronounced fear on the part of these animals. So again, this would be stress-enhanced fear learning, but you're seeing this other symptom here that's common with PTSD, which is this enhanced startle response, often taken as a measure of hypervigilance. Okay, so I said how, in behavior therapy, exposure therapy is not always perfectly successful against post-traumatic stress. And I want to show you what we've done with that, and for a couple of reasons. So our animals are going to get 15 shocks or no shock controls, just as we did before. But then we're going to bring them back to the stress context where they got shocked. We're going to do that for 5 days. They go in there for a session each day, but no shock, nothing's going to occur. That's half of the animals get extinguished and half of them don't. So there's no data for the home cage animals. They're just sitting in the home cage during this time, but here are the animals that are getting extinguished. So obviously the animals who weren't stressed, who didn't get shocked during this time, don't freeze during the session. The animals who got the 15 shocks show very pronounced conditioning. You expect that. They've got 15 shocks there, so they should show a very strong fear response, and they do. But as we extinguish them, bring them back each day from this, they're brought down to baseline levels. They show no more fear of that context. Then we're going to put them in the other context, give them a single shock, and look at their stress-enhanced fear learning. So what do we see? Well, here are the animals who sat in the home cage. This is just like you've seen before, except we've now introduced the delay, this 5-day delay between the stress and the shock because the other animals were being extinguished. So here's the normal fear you'd show after one shock if you weren't stressed. Here's our stress-enhanced fear learning that we've seen before. Here are the animals who completely extinguish the fear of that context. They're no longer afraid of this context, and yet they show the same degree of stress-enhanced fear learning, even though they're not showing any fear. So that's another reason why I say this isn't generalization of the fear of this context to this context. We can extinguish the fear, we can show there's no generalization there, and yet we still have this. And that's why I say that behavior therapy here is clearly working. It's getting rid of this associative fear, but it's not affecting this non-associative fear. That remains completely intact as if the therapy had never been given. And so this is the part that I think is why this component, I think, is exposure therapy is not quite relevant for. So I want to show you a really profound example of this, and that shows that it's really to try to push the envelope that this is not dependent on associative memory. And what we're going to do here is we have animals that have cannula inserted so that we can give a drug, APV, which is an NMDA antagonist. APV is your classic antagonist of the NMDA receptor. The NMDA receptor is the glutamate receptor that's responsible for associative learning. It has this property that allows long-term potentiation when a mild glutamate signal comes in and paired with that cell spiking. And that's what drives the plasticity that supports normal fear learning, and it's critical for normal fear learning. We're going to give to the hippocampus the memory structure that's important for declarative memory, explicit memories. We're going to give APV to the hippocampus in half of animals. The other half will get just the vehicle injected. They'll get their 15 shock stress. I think we can look at the data here. And so here's what happens when we bring these animals back to the context they got the 15 shocks, where they should show an associative memory. So non-shock animals during this time, shocked animals who got the vehicle, of course they're going to freeze here. They got conditioning to that context. They freeze tremendously. However, if the animals got APV, this NMDA antagonist, you see we can block the associative conditioning. And we've shown that a number of times before, that the NMDA receptor is really critical for this associative learning. But here we block the associative conditioning here. Now what happens when we give one shock in this other context and look for stress-enhanced fear learning? Well, here are the data. Here are the vehicle animals, so we see our stress-enhanced fear learning as we've seen before. Here are the animals where we block the associative memory. Completely equal stress-enhanced fear learning. They show, despite the fact that they have no memory for what happened here, when we ask them, did anything happen here, they're not afraid there. We see the same amount of this. And this is why we say it's independent of associative conditioning. It's really a non-associative effect. Despite the fact that they don't have any explicit memory for what happened, they show this kind of effect. And there are case studies of people, some of these are people who have been buried in construction accidents, actually suffered hippocampal damage, show no memory for what happened. You can bring them back to the situation, they still have no memory for what happened, yet manifest all the symptoms of post-traumatic stress disorder, including nightmares and all of that. So there's something going on here that's beyond the explicit memory of what happens. And it just emphasizes that this is very different than the associative memory that also happens when you've had a traumatic event. I want to push this way and even a little bit further in this developmental model. We took rat pups at 19 days of age and gave them the 15 shock stressor. There are no shock controls as well. The reason we picked 19 days is this is before they have any hippocampal learning. They can't do contextual fear conditioning at this time. It's just too young an age. The neural systems for that are not yet developed. We're going to wait 2 months here before we test them again, and we'll test them back in this context here to see if they have a fear of that context. And so, yeah, let's go there. So when they come back to this context here, they don't show any memory. These animals are not freezing. And this is, again, consistent with work that others had done developmentally, saying, oh, contextual fear doesn't develop if you have the conditioning at this day. We're just showing that even with our strong stress procedure, they don't develop contextual fear. They don't have a contextual fear memory. Here, when we waited 2 months, they're now young adult animals. We're going to bring them into the other context, and in that context, we're going to give them a single shock and see what happens if we get any stress-enhanced fear learning. So here are the data for that. These are the animals who didn't get any shock during that, when they were young, and so they're our non-stress controls. So this is the conditioning supported by the single shock here. But the animals who got this stress at this very young age show no memory for what happened, still have this greatly enhanced fear. So this early life trauma is setting these guys up. We don't see a fear response, right? They're fine when they go in there, and yet now they have a trauma and they're conditioned disproportionately to what they really should be happening here. Andrew Poulos, who did this study, ran a number of tests. So in other groups of animals, he looked for other signs and symptoms that happen when they've had this early life stress. And again, we're waiting 2 months here, so they're young adults. He put them in a standard test of anxiety. So these animals don't have the one shock. They don't have the test of this environment. Nothing's done over these 2 months to them. But they are now put in an elevated plus maze. So it's a raised cross, and half the arms are, there's a little barrier. The other half, they're out on the open, so they are afraid they might fall over. And what we look at here is that animals who are anxious spend more time in the closed, the protected arms, than they do in the open arms. What do you see when we look at this? The animals had the prior stress show much more time in the closed arms, much less time in the open arm, a classic anxiety profile that's used in rats. So they do have anxiety, heightened anxiety that's accompanying this stress that seems to last, again, for this long period of time. Andrew also took a number of other groups to look at the diurnal cycle and corticosterone. So corticosterone is the rat analog of cortisol. We know that it has a nice specific circadian rhythm. And these are all separate groups of rats. So we're looking at basal cort over the diurnal cycle. Here are the animals who didn't receive the stress, and they show the standard peak of just when dark occurs, so the sort of opposite of humans. They show their peak in cort levels, and then it goes down. So this is your normal cort diurnal rhythm that you normally see. Here are the prior stressed animals. What they have is a disturbed rhythm in the cort rhythm, right? So they show actually a 2-peak response. Depending upon when you look at them, if you look at them here, they're actually showing a reduced cort level. But you're looking at these, they have a higher level. It's not the actual level of cort, it's not just heightened, it's actually a disturbed rhythm that we're seeing in corticosterone in these animals. Andrew also took several brain structures from these animals and looked at the glucocorticoid receptor. So that's the receptor that corticosterone is going to be acting on. And we don't see any differences in prefrontal cortex. We don't see differences in hippocampus. But in the amygdala, we have this this increase in the glucocorticoid receptor. So more receptors for the corticosterone that's there. And again, this is a different cohort of animals than those others. So I've given just some of the properties of this stress-enhanced fear learning phenotype. So just to sort of summarize that is stress will increase subsequent fear conditioning. This is long-lasting, so I didn't show you data, but I showed you one study with the developmental where we waited 60 days between the stress and the treatment. Here, we've done other studies taking in an adult animal where we've waited 90 days after the stress, and there's no diminution over that time. That's the longest we waited. Remember, you get a diagnosis of PTSD if it persists over 30 days, over a month. This is 3 months, and it's a rat, so it's quite a long period of time, and we see no diminution. So it seems to stay with you forever. It resists extinction. Again, this idea that this is a component that's not successfully treated by exposure therapy. It occurs despite the amnesia for the trauma. One thing I didn't show you is the stress. We really think it's a change in the structures that support fear learning. I'll show more evidence. If we give the single shock first and then give the stress, we don't see a change in reaction to the single shock context. That's mild stress. It has to occur after the more traumatic stress has occurred. I think all of those things sort of tell you that this is not associative memory. It's really, I think I've lost my... It's not associative memory. It's really, and it's not generalization of an associative memory. It's really something different. We didn't see prior to the shock, we didn't see any freezing in that context. It occurs whether the one shock is paired with a tone or context. I always did context conditioning. We can do it with a tone as well, and we'll see enhanced auditory fear conditioning in there. It's dissociable from fear of the 15 shock context. We can make them amnestic for that. We can do a developmental amnesia. We can give extinction of the original 15 shock context. And it's accompanied by... I did something very wrong. Help.
Don't be afraid.
There we go. It's accompanied by increased anxiety, a change in the normal rhythm for cort, increased glucocorticoid receptors in the amygdala. I didn't show you data, but there's increased voluntary ethanol intake. There's increased floating in the forced swim test. The forced swim test is done as an animal model of depressive-like symptoms, and they show an enhanced depressive phenotype in that, and increased reactivity to a startle stimulus I showed you. So now what I want to do is turn to what has to happen during the stress to induce this phenotype. And given the change in glucocorticoid receptors and the amygdala, I want to focus on both the amygdala, and here I'm really targeting the basolateral amygdala complex, so the lateral nucleus and the basal nuclei, and corticosterone. So in this experiment, what we're going to do is give the same basic procedure, but we're going to give them a systemic injection of various doses of metyrapone. Metyrapone is a corticosterone or cortisol synthesis inhibitor. It blocks the synthesis of cort. and we'll, after this 15-shock stress, we'll take a blood sample and look at cort levels. We'll test that context. But we'll also give them a single shock, and this will be, again, the critical data, single shock to see if the fear learning is enhanced. So we look at cort levels. This is the blood cort after that 15-shock stress. So obviously the animals who received the 15-shock show higher cort levels than the animals who didn't get the stress. But what you see is that this stress-induced release of cort is blocked by metyrapone, the corticosterone synthesis inhibitor. So as you would expect, we've successfully blocked the elevation in cort produced by the stress. Now we'll just skip to the test data. So again, these animals got the single shock, and then they're tested in this context. So here we have the animals who didn't get the metyrapone. So this is just replicating. Here's our stress-enhanced fear learning. The animals who only got the one shock, not the stress. And this is what happened to the conditioning by the one shock and the stress animals, so what we've seen before. And what we see is a dose-dependent reversal. At this high dose, there is no stress-enhanced fear learning. So the phenotype depends upon the cort that's being synthesized during that stress session. If we look at cort in these animals, and I want to point this out, if we look at cort in these animals after this test, right, after showing this massive enhancement, we don't see significant differences in cort at that time. So at this point in time, cort levels, the elicited cort level by going into this context is not reflective of the symptom. I think there's a lot of controversy of what happens in cort in PTSD. And that's not what we're seeing is important at the time of expression. It's really, do you get this, I think this big increase during the stress itself seems to be what's the critical variable as it relates to cort. So if this is this effect of metyrapone on cort, and if it's acting via cort, then if we should be able to rescue the phenotype if we co-administer cort along with the metyrapone prior to the stress. The other question we have is are the cort changes themselves sufficient for producing the stress-enhanced fear learning? And if that's the case, if we just give cort alone without stress, can we mimic the pattern that happens? And so what we're going to do here is just as before. Why not? There we go. If we give, again, a high dose of cort or no cort prior to the stress, just as we did before. But what we're also going to do is just before the stress, administer systemic cort at a 10 milligram per kilogram dose or not, and then give the 15 shocks. I'm going to skip out here. We give them the single shock, and I'm going to skip out here to what happens during the test. So these are the animals who never got either of the compounds, no metyrapone, no cort. So here's our standard stress-enhanced fear learning. Here are the animals who got the high dose of metyrapone but no cort. And we see again that the phenotype is blocked. The stress-enhanced fear learning is blocked by metyrapone. Here are the animals who got this effective dose of metyrapone and we co-administered cort. We rescue the phenotype. So clearly the metyrapone effect is mediated via cort. Here are animals who did not get stress, only got the cort. And what we see is, and here are the animals who got stress, but here are the animals who didn't get any stress, only got the cort. It doesn't duplicate the effect. So cort alone is not sufficient. It's necessary. If cort is not there, then we're not going to get the phenotype. It can rescue the phenotype. It's permissive, but it's not sufficient to induce. So you need something more than just the cort to produce the effect. So is cort acting on the amygdala? So what we did in this experiment was to block those glucocorticoid receptors in the amygdala. So we have animals that are implanted with cannula into the basolateral amygdala. We're going to give the corticosterone, the glucocorticoid receptor blocker, mifepristone, at a half a microgram dose or just the vehicle prior to the 15 shocks. Give them their single shock, and then do our context test. Here are the data, and again, cannula placements in their basolateral amygdala. So here are the stressed animals who don't get anything injected into the, except the vehicle injected into the amygdala, so this really high level of fear. There's, the stress-enhanced fear learning. If we give them the mifepristone, so we block glucocorticoid receptors just in the amygdala, we reverse the phenotype, so it's clearly court acting on the amygdala, and it brings it down to the level of fear of the unstressed animal. So we clearly have an effect of court acting upon the amygdala here. So I said court is not sufficient. Something else has to also occur. What is that other thing? These are very preliminary data, unpublished, but I think they're interesting in that what might be the other thing. And you turn to, what do you think naturally would you jump to if you're a neuroscientist as working along with court to induce the effect? You'd think of some neuromodulator, right? Most of you would think of norepinephrine, but norepinephrine isn't it. We've never gotten norepinephrine to do anything in this procedure. So again, they get the 15 shots prior to the experiment. They had cannula in the amygdala, and we're going to inject the muscarinic cholinergic, so it's going to block acetylcholine, muscarinic receptors for acetylcholine within the amygdala, or they get the vehicle injected there. Again, they get a single shock, and then we're going to test them for stress-enhanced fear learning. So here are the data here. The animals, here's the non-stressed animals who got the one shot. We see the stress-enhanced fear learning, and scopolamine completely blocked, during the stress, completely blocked the stress-enhanced fear learning. So our hunch now is that it's somehow acetylcholine, which we know is released in response to stress as well in the amygdala, combined with the court that's producing the effect. And that's what we think is going on for the induction of this effect. So just again, it's a muscarinic cholinergic antagonist. Will prevent the development if just given during the stress. So to summarize kind of the mechanisms of the induction of stress-enhanced fear learning, court synthesis is necessary, but it's not sufficient for the induction of stress-enhanced fear learning. Court levels at the time of expression do not appear to be relevant to cephal. We've given the tyrapone at every other time, prior to the one shock, prior to the test. It doesn't do anything. It has to be given prior to the stress to work. So it's not a treatment because you don't know when you're going to run into trauma, or you wouldn't go there, right? So it's not an effective treatment for it. But and it's really what's happening with court at the time of the stress that's critical, and these other times are not so important. Court's action is mediated by the basolateral amygdala. We say that because of the ability to block glucocorticoid receptors only in the amygdala, blocking the effect. And cholinergic activity in the amygdala also seems to be necessary for stress-enhanced fear induction. So now I want to turn to the mechanisms of expression, and I want to focus on excitatory neurotransmission here. And if we just go back to this experiment that you'd already seen with the tyrapone, 2 weeks after we completed the experiment, we took the brains of these animals. So nothing happened for 2 weeks after the experiment, and we took the brains, we did amygdala punches, did Westerns to look at proteins for subunits of excitatory of glutamate receptors. So glutamate's your major excitatory neurotransmitter. Glutamate, just to give what'll become very important background, glutamate works on two major classes of receptors postsynaptically. So you have a presynaptic cell coming into the amygdala that's going to release glutamate. It's going to act on two different classes of glutamate receptors. One is the AMPA receptor. The AMPA receptor is your normal receptor for excitatory transmission. When it sees glutamate, it opens up a sodium channel that can excite the neuron. If enough sodium comes in, the neuron fires and sends a downstream signal. So that's your classic excitatory neurotransmission, is the AMPA receptor. The other type that is also acted upon on glutamate is the NMDA receptor. The NMDA receptor works very, very differently. The NMDA receptor is doubly gated. When it sees glutamate, unlike the AMPA receptor, it doesn't open up its ion channel. It doesn't respond to just glutamate. The NMDA receptor sits there, and it has to see glutamate, but the neuron also has to fire. It has to depolarize. That relieves a block of the channel and opens up the channel. And so the NMDA receptor is sitting there saying, do I see glutamate? Does something important happen to my neuron? Because the neuron is becoming depolarized, it's firing, and only then does it open its ion channel. And unlike the AMPA receptor, besides the double gating there, it also allows calcium to come in. What does the calcium do? It induces gene expression, induces the plasticity that's able to potentiate that synapse. How does it potentiate it? It creates more AMPA receptors to put into that synapse so that now when a glutamate signal comes, there's more AMPA receptors to let in sodium to drive that cell, so that you've potentiated that synapse. And we know for fear learning, for many types of learning, those NMDA receptors are critical for learning. Why? Well, they can tell two things are happening. A signal's come in, but something important has happened. Let's change the synapse now. AMPA receptors don't do that. They're just activated by the glutamate signal. So in everything I told you, I probably convinced you that what I'm going to see is a change in NMDA receptors, right? Well, here's the data on that. When we look at the amygdala, we're looking at the NR1 subunit of the NMDA receptor. And so the GLUN1 subunit. This is obligatory. To make NMDA receptors, you need this protein here. You need the protein made by this gene, the GLUA1, the NR1 subunit. And what you see here is our stressed animals. They either got metyrapone or not. There's absolutely nothing there, right? So it's not NMDA receptors. That's weird. Why is that? That's what we were hoping. But fortunately, we also looked at the AMPA receptor subunits. Those are the ones that do normal excitatory transmission. Typically, AMPA receptors are made of two subunits, a GLUA1 and a GLUA2 subunit. They put them together, and they're both there. The two of them together make your AMPA receptor. So what happens when we look at GLUA2, one of the critical AMPA receptor subunits? Absolutely no change, right? That's not changed either. So, Fanselow, what's happening? You're not finding anything here. Where we saw change was in the GLUA1 subunit. Okay, so this is the GLUA1 subunit, and what we see is in the animals who receive stress compared to the unstressed animals, we see this upregulation in GLUA1. Now, this is pretty amazing. Here's where the stress occurred. They went through all of this experiment, and then we waited 2 weeks, and we see this elevation in GLUA1. Nothing happened during these 2 weeks, right? So this is looking like a pretty long-lasting thing. Here's the other critical thing about this finding. The metyrapone given at this point that blocked the stress-enhanced fear learning also blocks the upregulation. Okay, so this really seems to be involved in some way. Now, it's also true that these animals had a lot going on. They not only had the stress, they had all of this testing and the single shock, and all of this could have done something to cause this long-term regulation. So what we're going to look at is animals who only get the 15 shocks or not, don't do anything, but then take at various time points the amygdala, look at this GLUA1 protein and see how it changes. And so here are the data. These are control data, mean and standard error for the home cage controls. And if we take amygdala immediately after the stress, it's elevated. A day later, it's elevated. 6 days later, it's elevated. 2 weeks later, it's elevated. So we see the sustained elevation in GLUA1, which is pretty unprecedented. People often see after a learning or a fear conditioning episode an immediate small change that goes away very quickly. In this procedure that's producing these very long changes, we have this very long upregulation of GLUA1 that's lasting, even without anything happening for this really, really pronounced long period of time. So just to give an interim summary so far, no changes in the NMDA receptor, no changes in the GLUN1, no changes in the GLUA2 subunit of the AMPA receptor. But these, what we did see is that this protocol that causes the stress-enhanced fear learning increases this long upregulation of GLUA1, and metyrapone blocks it, so it's following the stress-enhanced fear learning. So there seems to be a court-dependent increase in GLUA1 that happens in the basolateral amygdala after this treatment that induces stress-enhanced fear learning. So what's going on? And we have to come up. So now we get to the final moments where I get speculative, but this is, I think, pretty exciting, and so I needed to share it with you guys. And so here's the crazy hypothesis, that stress leads to some aberrant, non-hebbian. By hebbian, it means that you don't need these two events. You don't need both the presynaptic neuron and the postsynaptic neuron, the receiving neuron firing, right? The hebbian is when both of these things occur together that you get the increased potentiation. This is a non-associative sort of plasticity is happening in the amygdala. Why do I say this? What leads me to this idea? So there's a few things here. Again, I told you that AMPA receptors are typically a heteromer made up of both the GLU-A1 and the GLU-A2 subunit. The stressing causes an increase in GLU-A1, but importantly, not an increase in GLU-A2. So you have this part, but not that part of your normal AMPA receptor. We've shown, and others have shown, in a mouse, if you knock out the GLU-A2 subunit, what the animal does, what the neurons do, is to make AMPA receptors entirely composed of GLU-A1. So they make a monomer of only GLU-A1. Now instead of GLU-A1 and GLU-A2, I got only GLU-A1s. I'm going to put those two together. So what we're saying, and here's the little bit of a stretch, is maybe because we have an increase in GLU-A1 but not GLU-A2, we're making more of these GLU-A1 only AMPA receptors. Why is that important? If you make an AMPA receptor with only GLU-A1, if you lack the GLU-A2 subunit, suddenly the AMPA receptors become calcium permeable. It's the GLU-A2 subunit that gives the AMPA receptor its property of excluding calcium. When you make an AMPA receptor that's only GLU-A1, it allows calcium to come in to that. What happens, so in the GLU-A2 mouse knockouts, we see this great increase in long-term potentiation. Why? Because when glutamate is released on the neuron, if that neuron fires, it may open up its NMDA receptors, but in addition, those AMPA receptors, when they see glutamate, let calcium in. Also, even if the neuron doesn't fire, remember, the AMPA receptor is going to be activated by glutamate regardless of whether the cell is depolarized or not. If it's seeing glutamate there, it's going to open its ion channel, but now that channel is calcium permeable, just like the NMDA receptor. That's going to induce plasticity and cause potentiation of the synapses, but it's a non-associative. It's a non-hebbian form of plasticity, so a very aberrant form of plasticity there. So our hypothesis here is this traumatic stress may lead to an increase in GLU-A2 lacking AMPA receptors. And if you're enhancing non-hebbian, this non-associative plasticity, this seems like an explanation of why you're going to get very non-specific, very generalized learning, fear learning occurring. These neurons in the amygdala that normally support fear learning are going to be driven anytime they get any even mild sensory signal in there, be driven to induce plasticity, increase their synaptic strength. This may account for something like we observe in stress-enhanced fear learning. So next thing is to ask the question, are these calcium-permeable AMPA receptors in the basolateral amygdala necessary for stress-enhanced fear learning? Normally, they're not going to be there in any kind of real numbers. Normally, if you try to block them, you're not going to see much of anything. So what we're going to do is to see if we can block calcium-permeable AMPA receptors, if it can reduce the sensitized fear. And so we're going to use a specific drug that simply only blocks these calcium-permeable AMPA receptors, right? It only blocks the GLU-A1 only AMPA receptors. We're going to infuse that into the amygdala, so animals get cannula implanted into the amygdala, and then they're going to get the 15 shocks treatment here. Then right before they get the single shock, if it's these calcium-permeable AMPA receptors that let the shock do this disproportionate amount of conditioning, then if we block these calcium-permeable AMPA receptors with IEM prior to the single shock, we should be able to reverse that phenotype. So what happens when we test those animals after that? Here are the data. So here, it's kind of cut off there. So these are the non-stressed animals. They're not showing much in the way of conditioning. Whether they get, IEM or not doesn't make a difference. Here are the stressed animals who got the 15 shocks here, got the single shock. They got the stress, they got the single shock. And they got vehicle injected, so no IEM. Here's the stress-enhanced fear learning you see all the time compared to those. However, if we block these calcium-permeable AMPA receptors at the time of the single shock, we get rid of stress-enhanced fear learning, strongly suggesting that these calcium-permeable AMPA receptors are indeed important for generating this kind of phenotype. So we wanted to get this in another way and in a more specific way. So what we did is we generated, remember this we're blocking this receptor. It's already been created. We're blocking this receptor at the time that we're giving them the mild stress, the exaggerated reaction. What we want to see is can we do something after the stress to prevent the development of this. So what we did is we generated an antisense. So it's in a series of oligonucleotides that are in the reverse sequence to GLUA1. That's going to prevent the translation of the gene. So we're going to prevent the upregulation of GLUA1 by giving this antisense. And so this is just really to show it works. When we give our antisense, we'll see less GLUA1. A missense is just the same sequence, same oligonucleotides, but they're in a scrambled sequence that makes no sense. It doesn't code for anything. So that's the control injection. So we can see that our antisense works. That's all it shows, is that it does block the translation of GLUA1. So we're blocking the formation of the protein. And so what we're going to do here is, again, implant cannula into the amygdala, give animals their 15 shocks. Then after, we did this a couple of ways, either immediately after the stress or before the one shock or both, we'll give them an infusion of this GLUA antisense, missense, or the saline vehicle. Then we'll give them a single shock in this context and test for stress-enhanced fear learning. So here are the data. Here are the stressed animals who got just the vehicle injected. So here's our high level of stress-enhanced fear learning. Here are the stressed animals who got the missense. Again, we have stress-enhanced fear learning. Here are the animals who got they got the stress, the 15-shock stress, but they got infusions into the amygdala after the stress and before the one shock of the antisense, and we reverse the stress-enhanced fear learning, bringing it pretty much down to the level of unstressed animals. This is the first thing we've ever been able to do after stress that seems to prevent the development of the phenotype. Should be something here. We also, if we just give one dose, so we give just a dose after the 15 shocks, we also are successful at blocking the phenotype, so we didn't have to give it both times. Then what we did, let's see, gotta push that. What we did is to wait 7 days after the stress, bring them back and give them another test. Now, one thing is that by giving antisense downregulates typically for about 12 hours. Okay, so it's a pretty short lasting. This is going to be well beyond any effects of antisense. Does everything just return after the antisense is clearly worn off? It shouldn't even be active here, and we're seeing an effect, but how long does it last? Well, when we bring the animals to that context a week later and give them the stress, just test them, we're not giving them any more stress. What we see is we've still reversed the phenotype. So this seems to be a long-lasting elimination of the development of that phenotype from occurring. So we're pretty excited about that result because it's the first thing we've been able to target that really seems to permanently reduce the phenotype. Now, we took these animals and we asked them, did you forget your fear of the original training context, or are you still afraid of that context? And ask them that question, right? So we just bring them back after this, is that fear there? And clearly it is. So these are the non-stressed animals. These are the animals who got the 15-shock stress and the antisense. We did not cause an amnesia or a loss of memory of the threatening place. So that's still intact. I think that's good. Something really bad happened here. You should probably remember what happened really bad. Yes, get rid of these disproportionate fears, development of those, but learning that something bad happened is probably a good thing, and that memory is still intact. It means the amygdala is probably functioning fine. You just prevented the development of this. And then what we did is we said, well, if the amygdala is really fine, if we, without any antisense now, give them another bout of 15 shocks, can we induce stress-enhanced fear learning, or do we permanently muck up the works in the amygdala? So we're going to give them now another bout of 15 shocks and test them. And what we see is we can induce it again there. So the amygdala is functioning fine. We haven't lesioned it or anything like that. We've just eliminated the effects, the detrimental effects of this prior stress. So it did not eliminate the fear of the truly dangerous place. It did not permanently alter amygdala function. So our working model of stress-enhanced fear learning is that we get this severe stress. It works both to cause the release of corticosterone in the rat, what I looked at, or cortisol in the human. It also is acting through acetylcholine release on the amygdala. Those two converge, and what we're going to see with that convergence is an increase in glucocorticoid receptors and also an increase in these calcium-permeable AMPA receptors. And that leaves you in a situation that now if you receive just the mild stressor, it's going to be acting on an amygdala that has these calcium-permeable AMPA receptors, leading to this enhanced fear conditioning. So that's our working model of what's occurring. So just some final conclusions. Induction of stress-enhanced fear learning. Like fear conditioning, standard-associated fear conditioning, stress-enhanced fear learning is dependent upon the amygdala, and we think one of the things that happens is we're changing the basic associative machinery and how that works. So now the amygdala is more reactive. It's set up to be more responsive to future stressful events, even events that are not that stressful, that are relatively minor stressors. Corticosterone plays a role, acting on the basolateral amygdala, plays a role in the induction, but not the expression or the consolidation of stress-enhanced fear learning. But court alone is not sufficient. We think that perhaps that acetylcholine release has to work in conjunction with the court in order to produce the effect. In terms of expression, there's a court-mediated increase in GLUA1 of the AMPA receptor, but not GLUA2 subunit of the AMPA receptor. This increased expression of GLUA1 is necessary for the expression of stress-enhanced fear learning. So we think the induction of this non-associative, this non-hebbian plasticity mediated by calcium-permeable AMPA receptors is the culprit in all of this. And we were very surprised and fortunate to find out that I think with this GLUA-anti-1 sense, which only has a temporary effect on GLUA1, really seems to permanently reverse the effects of the stress while leaving the amygdala intact for what it's learned already and protective fear responses. So we think that there's some kind of self-maintaining process, that once the GLUA1 is upregulated, it self-maintains in some way. I think that if we figure out what's going on with that self-maintenance, we'll have a lot of targets, a lot of keys of what to hit with this. But so are we working our way a little bit, couple of baby steps closer to a potential cure? Well, if you think about what this is doing, obviously, surgical administration of antisense, I always want to say this, is not we're not advocating this as a cure. This is too invasive a strategy, I think, to use. But it has a lot of the effects we'd want as a cure. One is a single treatment with this permanently restored normal fear function. The amygdala was functioning, and it was functioning normally after that time. So it's not a drug that you have to constantly take forever to suppress a symptom. Rather, it seems to be able to restore the amygdala to its pre-traumatically stressed state, which is, I think, what we'd want in a cure. Learning appropriate fears is very adaptive. Fear evolved because it protects you from things. And so not erasing your associative, your meaningful fear, and leaving the amygdala able to learn fear, I think, is a really critical thing. And this seems to do that. And so obviously when we want to try to work on, and what we're trying to work toward now, are how can we develop non-invasive strategies that'll stop this either maintenance or the upregulation of these calcium-permeable AMPA receptors, and maybe that will be our help. And I also want to point out that I'm saying this non-associative component is really, really important, and it's been ignored. We don't have a treatment yet for that. But I'm not denying that there are clearly important associative aspects. We have targets for that with exposure therapy, behavior therapy. We have ways of trying to make that better, and that's definitely an important goal. But I think these two things have to advance together if we really want to make things better for the people who suffer from this. And I'll end with that and just thank a lot of people who really contributed to this work over the years. I'm especially pointing out Andrew Poulos, who did some of the work I showed at the beginning with the developmental model and the really enhanced reactions to mild fears, and Jennifer Pericini, who did all the work with the antisense and the metyrapone and that work in the amygdala. But it was a big team of people who really helped with all of this. Thank you.
Thank you very much. Thank you very much. We're going to take a 20-minute break for folks. Grab a coffee.